All right, folks, so we're here to talk about Chapter 21. This is Part B of Chapter 21, and in Chapter 8, we discuss the innate immune system, and in Part B, we are now discussing and moving on to the adaptive immune system, right? So this is, again, innate talks about the first and second line of defense, and really the adaptive discusses the third line of defense, and that's what Part B is really all about, is looking at the defense. Okay, so when we look at the adaptive immune system, the key word that you need to be aware of here is specificity, right? Otherwise known as something being specific, okay? The adaptive immune system is a specific defense system that will eliminate pretty much anything abnormal in the body or anything pathogenic, right? So, for example, if we have any types of pathogens that we have in the body, being, for example, bacteria, okay, and viruses, okay, both of which are, again, considered pathogens in the body, we have a specific then defense against those, or an attack, right? So, again, we are not, the immune system's not about attacking something already there. It's about that one thing invading our body and then us responding to that, right? So it's a response, and this is going to be a specific response. It's not a general response, but a specific response, and really that's what the third line of defense is, being specific or specificity, right? And so how does it do that, right? What's the action involved in doing that, right? It will amplify in an inflammatory response, right? And activate what we call a complement, right? And what do we mean by amplify? What is the word amplify, right? Another word to talk about when you talk about amplify is what? Increase, right? Increase the inflammation response, right? And as we saw in Part A, the inflammation response is really quite important, right? And if we can increase that, again, that's a fine line, but generally speaking, if you can increase inflammatory responses, then that's going to be a better type of defense towards the particular pathogen or the abnormal cell in the body. And when we talk about complements, we'll discuss that in a minute, right? So complements also include, for example, antibodies, and we'll talk about that as well. So those are two big advantages of an adaptive immune system, okay? That again, it amplifies an inflammatory response and activates complements. It's specific, all great benefits to it, but there are actually some disadvantages, right? Some shortcomings, if you will. Disadvantages to the adaptive immune system, right? And so the adaptive immune system, when we talk about the adaptive immune system and its shortcomings, one of the biggest shortcomings is that it has to be primed, right? By an initial exposure, okay? And priming does take time, and that's really what its shortcoming is. And what do we mean by primed, right? Prime means, another word to talk about that is to start, right? Or otherwise known as initiate, right? It has to be initiated, right? And we have to initiate it by the initial exposure, right? So again, part of the adaptive immune system, again, when we go back to first and second line of defense and third line of defense that we're discussing, we know that the first and second line of defense are by nature, right? It's natural or innate, right? That means we're born with it, right? Yes, it gets stronger. That's absolutely true. But yes, we're born with a basic natural immune system. That's the first line, second line of defense. But the third line of defense is, as I mentioned in the first part, it's learned, right? And how do we learn it? By exposure, right? And again, that's the only way we can actually gain the third line of defense is actually exposure. And that is a disadvantage because some of the exposures to certain types of pathogens can do carry the risk of and can have a risk of death, right? Or dismemberment, right? Or impairment of the body. And those are definitely some shortcomings there. But all in all, it has to be that initial exposure, so that way the body responds to that initial exposure by learning, right? And that's what we talk about when we talk about priming. Okay, so here's some characteristics of adaptive immunity. It's very specific, and then we just talk about, specificity, right? It's got specific antigens, and we're going to talk in good detail about specific antigens. Number one, right? Number two, it's systemic. Okay? It really means throughout the whole body. That's not just localized in one area, but the adaptive immune system is, its effects can be throughout the whole body, right? And it has memory, right? Mounts a memory, right? That memory, okay, which means our adaptive immune system is memorized the first exposure, okay, and then because of the second or third or fourth or et cetera exposures, it knows, because it's memorized, how to attack, right? How to attack, okay, the antigen, right, or the pathogen, if you will. Okay, so it has memory, and that's a really nice feature to have, because you can keep that memory for a very long time in your body, and it memorizes, if you will, how to attack that particular pathogen. Okay, so when we talk about the adaptive immune system, there's actually two branches, right? Number one is the humoral, and the second one is the cellular, right? And humoral, you always think of antibodies, okay? The humoral part of the adaptive immune system is when we synthesize and create then antibodies, right? And those antibodies go to work with a particular type of foreign invader, right? And cellular basically means cells, right? Cells, if you will. And these cells participate in the attack of the foreign invader, right? So this one has, again, we talk about antibodies, and in the second, the cellular are the cells that participate in direct attack, right? And we'll see that some of that's indirect as well. So we'll go into detail here with both of these. And so when we talk about humoral immunity, again, the key word is antibodies, right? Humoral immunity is about antibodies in the adaptive immune system, right? Who makes these antibodies? Lymphocytes, right? These lymphocytes then make the antibodies, secrete the antibodies from the cells, and those circulate freely, right? And they circulate freely in body fluids, okay? What are some examples of bodily fluids, right? Extracellular fluid, blood, and lymph, right? Sorry if you can't read that. Sorry, that means blood, lymph, and extracellular fluid. Those are what we mean by body fluids, right? And again, antibodies are free to circulate in all those areas. All those areas are found in all parts of the body, and again, produced by lymphocytes, right? So, how do they work in a short sense, right? They can bind to a target cell, right? They can inactivate the target cell, and they can also mark it for destruction, right? So again, they inactivate the cell. Sometimes we call this neutralization, and we'll see this in just a second towards the end of the slide, or excuse me, the PowerPoint. And neutralization is all about just inactivating the particular target cell or the antigen, if you will, and preventing it from causing more damage. And also what it does is marks it like a flag or an alert system, telling the rest of the immune system to attack that particular cell or that pathogen. Okay, and in cellular immunity, when we talk about cellular immunity, we're talking about the lymphocytes as well, okay? And so how do we do this, right? How do these cells that we call lymphocytes do it? We can do it directly by directly attacking the cell itself, or indirectly, right? And so how do we do this indirectly, right? So we kind of figured out directly how this works in part A, right? phagocytosis, for example, some of these can actually undergo this, right? And then indirectly, what do we mean by this? We actually, the lymphocytes can release chemicals, right? Those chemicals can enhance inflammatory response, or they can act as activators for other lymphocytes, macrophages. Some of those, for example, belong in the second line of defense, right? So again, we aren't just involving the adaptive immune system here, but also the innate immune system as well, right? Okay, so we talked a little bit about antigens already, right? We keep talking about this discussion of antigens, but what are antigens themselves, right? Antigens that are any type of substance, right? Okay, any type of substance whatsoever that can actually get the adaptive immune system to start mobilizing, right? And getting the immune system to elicit or create a response, right? All right, so any antigen whatsoever, or any substance that we consider an antigen, is anything that can get the adaptive immune system going, and again, get an immune response, generally speaking, right? Okay, most are large, complex molecules, normally not found in your body, right? We call those non-self. So if you remember self versus non-self. Okay, and we talked a little bit about that in part A. What is a self versus non-self? Where a cell of our own body, our immune system cells of our own body, patrol areas in our body and run into frequently our normal tissues and cells of our tissues. And if they recognize our cell as being self, they leave that cell alone. If they recognize them because of the receptors on the surface of the cell as a non-self, then they elicit an attack, right? And that's what we mean by non-self, right? Okay, so these chemicals do not have the receptors that our tissues or cells normally have. They're considered non-self, and because of that, they are, again, attacked, right? Okay. So antigens can be in several different forms, right? Some can be complete, right? Some can be what we call incomplete. We call those haptens. They contain antigenic determinants, and we'll see that in just a second. Or they can be self-antigens, right? When we talk about a complete antigen, if you will, okay, it has two really important properties, right? We talk about immunogenicity, right? The word immunogenicity, okay, comes from two words meaning immune system, means to start, right? Genicity comes from the word meaning genius, right? And that genius means to start or beginning, right? And what we're discussing here, in fact, okay, is talking about its ability, the antigen's ability to then stimulate proliferation of lymphocytes, right? Okay, basically kick-starting the lymphocytes to then create more cells for immune response, to attack or to secrete antibodies. All right, so that's what a complete antigen does. A complete and whole antigen will start the lymphocytes, kick off their immune response, okay, first one. And number two, reactivity, right? Those activated lymphocytes and those antibodies that get made and released then react, right? They react with the antigen, right? So it has the capability of immunogenicity, starting the whole process of the immune response, and number two, be able to react, right, to the things that are happening, right? Okay, and so when we see, for example, proteins, polysaccharides, lipids, and nucleic acids, different types of macromolecules, if you will, many of them are complete, right? And so we see these, for example, in pollen and microorganisms. They give you two great examples of those because those are where you find on their surface, if you will, macromolecules that are proteins, polysaccharides, lipids, or nucleic acids, all of which are all your four macromolecules. So it doesn't necessarily need to mean that an antigen is a protein, although many antigens are proteins. There's a majority of them that are protein-based. They can also be based on the other macro. And so what do we talk about when we mean, an incomplete antigen, right? These are also called haptens, by the way. So when you hear the word hapten, that's an incomplete antigen, right? It's a piece or a portion of what we call an antigen. And that's not to say that the antigen is broken apart into pieces. It just means it's part of a molecule, right? It's a molecule that's so small that by itself it's not immunogenic, right? That means if we put this into our body, okay, or on the surface of our body, it's so small that the immune system does not respond to it, right? So you must ask yourself, then why would this be part of the immune system? And what this means is it becomes immunogenic, really, if the incomplete antigen attaches itself to our own proteins of our own cells of our own body, right? Or the proteins that are sometimes secreted out in blood or in fluid, right? That are part of our body. So when they basically attach themselves to those proteins, the combination of both of those, right? The combination of the combo of the protein and the hapten together, okay, is seen by the body as foreign, right? So if I wrote A, and A equals the hapten, okay, this is molecule A and this is the hapten, and B is the cellular protein, okay, and I'll write PRO to represent protein, right? Okay, and in our example, when we get A into our body and the surface of our cell has B, that protein, and when they both bind, let's say A, and the line here represents the bond between A and B, they bind together on the cell surface, okay, and this B would be on the cell surface, then that equals, right, or that means then when the immune system comes around and sees that on the surface of the cell, it says attack, right? And so what it will actually do is not only attack the A and the B, but also what's attached to it, because it doesn't know the difference. And it will then start to attack and then destroy those cells. And why is that really important? Because a hapten that becomes complete on the surface of our cell tells our immune system that those are foreign, and then actually kills our own cells or destroys our own cells. And that causes damage, and that can actually lead to damage to the body, right? And so we see this, for example, in poison ivy. Poison ivy tends to have there's a kind of a lipid-based chemical that's found on the surface of the leaves of a poison ivy plant. And we see those, and when it interacts with the skin, it forms a bond between the skin layer, okay, and it actually soaks in past the epidermis layer and then forms bonds on the cells. And then what happens is the immune system will attack not only the poison ivy molecule, but what is binded to, and that's the cells. We have a red rash, breakout, lesions occur, and that's really our immune system attacking itself. And what we need to do is actually find a chemical, an antagonist that actually neutralizes that particular protein, and that's actually how you actually can relieve poison ivy, right? But animal dander can do that too. Some detergents can do that, depending on the person's body. Cosmetics can do that. So these are the four things that, when binded together, again, not as all together, right? All four of them together, but one of these binding to the cells of our body cause then an immune response, right? Okay. Okay, so sometimes we talk about these being, for example, an allergy, okay, or an allergic response, right, to some of these things. And that, but basically what that means is it's an incomplete antigen attaching itself to the cells of our own body, and then causing that combination, that binding of those two separate molecules together, causing then an immune response where the immune system cells destroy the cells and then cause a further inflammation response, and this is what you see as an okay. So when we talk about antigenic determinants, right? Antigenic determinants are parts of antigens, right, that the antibody or the lymphocyte receptor bind to, right? Okay, so this is the antigen, and this is the edge of the antigen where the antibody will attach itself to, or the lymphocyte will attach itself to. And we talk about this one because we talk about antigens having many different types of antigenic determinants or receptors that cause the immune response to happen, right? Okay, so we see this here, and I want to kind of take a look at this just so you know. Okay, we've got, you know, just one antigen here, right? And we've got 1, 2, 1, 2, and 3 determinants, right? These are portions of the surface of the antigen where you're going to have then the binding site to an antigen that's completely different, right? So we see that for number 1, we have antibody A that fits perfectly. It fits perfectly onto the surface of that antigen, that determinant. You have antibody 2 that fits perfectly there as well. And then antibody 3 that has a receptor that fits that antibody, which tells you that these 3, okay, antibodies are different antibodies. That means that our body can actually produce many different types of antibodies, which we'll talk about also during this PowerPoint as well. But we have different antibodies, some of which those antibodies can attack the same antigen. And that's what we're seeing because we have different types of receptors on one particular antigen, right? So here's number 1, here is number 2, and here is number 3. All part of one antigen, right? On the surface of that antigen. And again, all 3 different antibodies, right? Okay, so when we talk about self-antigens, right, we talk about this particular type of protein, MHC protein. And what an MHC protein stands for is the major histocompatibility complex protein, right? And it's a self-protein. It's a protein that's actually located on the surface of our cells, right? And each and every cell has these actual proteins, surface proteins on the surface of a cell. So again, I'm going to draw our typical cell, whatever this cell might be, right? And we've got these different, again, receptors, right? Located on the cell surface. Okay? Maybe say this receptor looks like this. Right? And that's our MHC proteins, right? Our major histocompatibility complex proteins, right? And so these aren't really antigenic. The reason why we're not talking about these is because, one, they're located in our body. So when our immune system cell, okay, located, let's say, our immune system cell is here, okay, and it's got a receptor like that, okay, and let's say there's more of them here, more of them here, more of them here. When it interacts with that receptor or those receptors right there, okay, it's going to recognize this cell as self. You are self. Okay? in our own body, right? In our own body. And so these MHC proteins, again, can also be part of determining our own body's cells, right? But again, if, for example, we took this cell here in question, here, and we stuck it, for example, in another person's body, well, their receptors and their immune system receptors are going to be completely different than ours. And we'll, again, let's say this is now the other person's immune system, and we've stuck, let's just pretend we've put this into another person's body. When their immune system cells interact with those receptors, that can equal non-self. And non-self is going to mean what? Attack. Okay? Now, where do we see this? Why are we talking about putting cells into other person's body? Well, we talk about that, and we're concerned about that when we see, for example, transfusions or grafts, right? Like blood transfusions or tissue grafts, right? So we got to make sure that those receptors, one, can be taken off, or two, are compatible with the other person's, the transferred person's body, right? And if not, we have an attack occurring, right? Cells will attack, the immune system will attack the cells, and then we have destruction, right? Or clotting can actually occur here, right? So again, those exist on surfaces of our cells, right? All right, so moving on, when we talk about the adaptive immune system, it actually involves really three types of cells, right? Okay, three different types of cells. Your B cells or B lymphocytes, your T cells, your T lymphocytes. The B cells participate in humoral immunity, and what that should mean to you, right? If you were to look at that, that should mean what? Antibodies, right? Antibodies. And cellular immunity, right? T cells participate in the attack, okay? The cellular attack, or the cell attack, right? Two types of lymphocytes. Those are the two cells that are important in the adaptive immune system, and another one we call the antigen presenting cell. Antigen presenting cells don't really respond to a specific antigen, but they play a really important role, an auxiliary role, okay, a supporting role in the immune system. We'll talk about those as well, okay? Before we talk about or move on to those cells, what I really want to look at here is actually where the B and T lymphocytes get made, how they get matured, how they develop and mature, and then how they get activated. Because that's really key to understand as far as how these adaptive immune system cells work, okay? So we first start off with the origin, right? Where they come from, if you will, where these B and T lymphocytes come from. And we know from previous chapters, for example, in the lymphatic system chapter and the beginning of the blood system, we talk about that the lymphocytes, right, blood, if you will, and those B and T lymphocytes all get made by the red bone marrow. And if you recall back from blood, there's a stem cell, hematopoietic stem cell, that actually will differentiate or turn into any type of cell, any type of red blood cell or any type of white blood cell, including these lymphocytes, either B or T, depending on our needs for our body. So our body's communicating with the bone tissue and the red bone marrow, telling them, hey, we need B cells, we need T cells, or we need other white blood cells, lymphocytes, eosinophils, basophils, platelets if you need them, red blood cells, etc., etc. But all of which start at the red bone marrow, right? That's the origin of both, you know, going back to B and T lymphocytes, where they come from, right? Where they're okay. So in this figure here that you see in the book, again, here's our red bone marrow, right? Here's our long bone, if you will, and we know that our red bone marrow occurs right here at the ends, right? Normally speaking, right? That's where our red bone marrow would be occurring there. Okay, and so we have the origin there. So the second part is maturation, right? This is actually growing up, right? Getting stronger, you know, growing up, becoming adults, right? Or trained, if you will, B and T lymphocytes, right? So lymphocytes, once they're made, they take an education, you know, period, right? And that's about 2 to 3 days, right? Okay, and they really occur in the primary lymphoid organs, right? And we know that to be the red bone marrow and the thymus, right? You recall that from blood, if you will, or the cardiovascular system. We know that the primary lymphoid organs are the red bone marrow and the thymus, both of which mature B cells and T cells, right? And so why do they mature? What's this maturation process? Why aren't they just ready to go right out the gate, right? Why can't they just be made at the red bone marrow and then they're ready to go and they're ready to be used? Well, one of the big reasons is actually because they must have education, right? They must have some kind of exposure to things to be educated, right? We talked about the adaptive immune system, one of them being exposure, right? And this occurs even in maturation, where we see the B and T lymphocytes being matured, right? Being educated, and the way they can be educated is actually exposure, right? And so when we take a look at immunocompetency, right? We see that the lymphocytes have to be able to recognize only one specific antigen, because again, the key word is, again, in all about the adaptive immune system, is specificity. Being specific, right? Being specific. So again, depending on what B and T lymphocytes get made, those get sent to the bone marrow, the red bone marrow and the thymus, and they're presented with, you know, an antigen, and they must then recognize that one specific antigen, right? So this is actually, number one is about training for a specific threat, right? That's what that means, right? That's our threat, our one threat, okay? Number two is self-tolerance, right? The lymphocytes must be unresponsive to our own antigens, the ones that we have in our own body, okay? So they're about training, right, for the non-threat, okay? That's really what this means here, right? Immunocompetence and self-tolerance, right? Okay, so let's pretend that this is a police force, and you know, this is a person that applied to the police academy, got into the police academy, spent some time, you know, in the police academy going through training. And one of the things is, what is a threat versus not a threat? What is a bad guy versus a not bad guy, a regular citizen, you know, of a town or a city, right? And who's the person that we need to, you know, assess the threat or arrest the person, right? What is considered against the law and what is considered not against the law. These are this is actually, very generally speaking, what these lymphocytes do in maturation. They recognize the one specific threat, and they actually look at what is not a threat, right? So they spend the 2 to 3 days looking at this, and that's really what, as simple as it is, that's really what's going on. Okay, and so you see the red bone marrow and the thymus here, okay, and the red bone marrow is where B cells do that maturation process, right? They do that education, if you will. That happens for the B cells in the red bone marrow, okay? And in the thymus, the T cells do their maturation process there. And that's why this is split up, right? The B and T cells have two places, different places where they mature, right, if you will. Okay, the third portion here is what we call seeding, right? And what do we mean by seeding? What does the word seed mean? Why would you seed something? If you were a farmer, you would seed a crop. What does that really mean? Well, basically you would, back in the old days before machinery, you could actually throw the seed out, right? Or plant the seed by hand, right? Or in modern times, you take a implement, and you take a tractor, if you will, and that tractor automatically, puts the seed into the ground. That's what seeding is basically all about, right? And if you think of that in this context, that's why we call this seeding, is because we actually take these B and T cells, right, after they've actually come out of the, let's say, their police academy in our example, and are ready to be on the beat, on patrol, in traffic, and on the streets, right? We call these naive, by the way, right? Okay? And just make it simple, these would be our little rookies, right? Okay? And all that means is these B and T cells haven't actually run in contact in real life with a threat yet, right? Those are what we call naive B and T lymphocytes. And what we do is we send those out. We seed basically them in our body, if you will, right? So we also use the word colonize, but that's actually what we mean, seeding, sending them out to the places in our body, so that way they can actually first interact for their very first time with the threat, right? And so where do we seed them? Where do we send them to? The secondary lymphoid organs, right? And what do we mean by that, right? The lymph nodes, our spleen, and other places, right? If you remember that, you got to go back to your lymphatic system, but secondary lymphoid organs, right? Mainly lymph nodes and spleen, but you have some other places as well. And this is where they actually have an increased chance of an antigen encounter. Okay? That antigen can encounter, by the way, can actually be hours, could be seconds once they've been seeded, or can be weeks, if you will, sometimes months or years that they've never seen it, but they stay around for the threat, right? So it just depends. And here in part 3, you see, that's your number 3, you see the seeding, right? Where do these get sent to, right? You see the thymus and the red bone marrow, you see the arrow kind of both melding in, and you see the nice lymph node, right? Where they get seeded into, right? B and T lymphocytes, okay? And number 4 is our antigen encounter, right? So those naive cells, right? Their first encounter, right, with that antigen triggers the lymphocyte to then change, right? And the lymphocytes will change, right? So once it interacts with that cell, right, it's going to change, okay? The lymphocyte differentiates, we call that the changing there, okay, into a active cell, right? And we only do that because it binded to the antigen. This process, we call this process here clonal selection, okay? By running into the antigen, they've been chosen, right? Or, nobody's chose it, if you will, but that's what we mean, that we've been selected, right? Because we've ran into the antigen to differentiate into an active cell, right? Okay? And this is what's occurring here in the encounter, right? You see these little antigens here interacting with the lymphocyte there, causing clonal selection, right? Okay? Clonal selection will occur, becoming activated, right? That's really what this means. Okay? So what happens once it activates? Once the antigen binds to the lymphocyte, what happens after that, right? The lymphocyte proliferates. What do we mean by proliferation, right? In a cell, that really means what? Right? Cell division, right? One cell becomes 2 cells. Those 2 cells become 2 more cells, right? And so on and so forth, right? So originally we had 1 cell, and now we have 4 of the same type of cell, and that's what we call about proliferation, right? The exact copies of themselves, and we call all of these copies here clones, right? These are our clones, okay? Clonal selection, those activated cells turn into clones, right? Now, some of these clones may be chosen as effector cells. What they do is actually directly fight the infection. They attack the particular antigen. And some of them, a few of them remain as memory cells, right? Okay. And so this is what we see down here, if you will. Okay. We see that these cells, when activated, start to become these new types of what we call clones, and you can see that there's, you know, now 7 there, right, in our example. And both B and T lymphocytes will do this. Both B and T lymphocytes will actually produce clones when stimulated by an antigen. And again, it's that antigen specific, right? Okay. So when we look at our immune system and we look at antigens and how they, you know, stimulate the immune system to do its job to then stimulate to make clones, and those clones do attacking or do the memorization, you know, how does this process really, you know, start? How does this process really, you know, where does this really come from, if you will, from our body, right? How do we know from the many things we interact with what begins, you know, an immune system response and what doesn't? And that really has to go back to your ancestors, right? To your, you know, your great-great-great-great-grandparents, to your great-great-great-great-great-grandparents, and the line all the way up to the very beginning of life, of human life. Those genes are, again, carried on from, you know, one person to the next generation to the next generation. But in fact, the genes that get carried on, some of them, you know, code for the antigens that we create, okay? And when we look at this, right, is actually, it's because those humans in some time of the history have interacted, right, with antigens in the past, right? And because of that, we've then created an immune response to it. And then our genes have changed. And then we carry those genes for the same type of immune response to those particular antigens, right? And that's really what this is all about. We know that our genes carry then the changes that we've experienced because we've interacted with these antigens, right? That's really interesting, right? Because we've got at least 25,000 different genes that code for about 1 billion different types of antigen receptors on the lymphocytes, right? 1 billion. It's a huge number, right? Billion different types of receptors, right? Okay, so again, it goes back to the genes, it goes back to your ancestors, right? And that's where we see certain folks can actually have a good immune response to a certain type of infection. Some people do not. And that's really because the genes can vary from person to person, from DNA to DNA. So that's where you see the difference there. So when we look at that, we call this positive and negative selection. Okay, so T cells, when they get presented, right, those MHC proteins of our own body, right, The ones that can recognize those are kept. The ones that can't recognize them, the ones that can, they read them and say, hey, this is, bad for you, get destroyed, right? So the thymus destroys the ones that are not trained correctly, right? Okay, so think of these as little robots, right? And if the robot gets tested and the robot doesn't do what it's supposed to do, doesn't recognize the MHC proteins, then the thymus rejects them, right? Kills them off, right? Causes them to undergo apoptosis, right? Which is what we know to be cell suicide, right? Which I mentioned before, right? Okay, programmed cell death, right? And that's what you see here, right? This is positive, negative selection in the thymus, if you will, right? Okay, so let's talk about the antigen-presenting cells, right? Antigen-presenting cells, right? Okay. So we've seen these before, right, folks? Right? You've seen what dendritic cells do and macrophages do, right? These are both of which are the second line of defense, by the way. I'm part of the second line of defense, right? And we know how those operate. We know these to be phagocytes, right? Those are phagocytes, if you will, both of those. And we understand the process of phagocytosis. We understand how they're destroying. They first interact. These are the first lines. This is the first, interaction of a particular antigen or whatever. And then we see that these, begin to undergo phagocytosis and break them and kill them. But one of the interesting things that they do is actually they undergo this process of being, if you will, antigen-presenting cells. So in fact, they break pieces up, right? So the cells that that they've destroyed, they take fragments or pieces of it, and they actually present these, okay? They take them to T cells, okay? And they show the T cell, they take it up to the T cell and show it to them, give it to the T cell, and basically saying, look, this is what we've destroyed, just so you know that this is in our body now. And then what it does, and the effect of that, is that the T cell will then activate. And that's really where the alarm system goes off, and the T cell will become activated and then cause then a cascade of immune system response, right? So really interesting that the cell itself presents it to the T cell, and then the T cell will activate, right? B cells also do that as well. So not only do B cells produce antibodies, which they do, some of those B cells actually are antigen-presenting cells, that they destroy they swallow up the antigen and then take that piece to the T cell, right? That's what we see here, right? Dendritic cells, we know that they're found in connective tissue, the epidermis. We find dendritic cells there. Okay, they mobilize, they patrol that area. Okay. Dendritic cells, when they find a pathogen, they phagocytize it, right? Then they take it to the T cell in the lymph node and present it to the T cell, right? And again, where do we find dendritic cells, right? They're part of the innate immune system. And what do we know what T cells are, right? They're part of the adaptive immune system. So we see this link between innate and adaptive, where they're both really connected to each other, where the innate presents to the adaptive and tells the adaptive, hey, this is what we find, so then we can actually elicit complete immune response, right? So here's a dendritic cell undergoing phagocytosis, right? See these little extensions of all the cell itself, right? These are what we call pseudopods. These pseudopods, stretch the cytoplasm out, stretch the cell membrane out, and then kind of basically stick or rope, if you will, pathogens and bring them into themselves, right? So we see a dendritic cell doing that. Right? Macrophages also do this, right? We find macrophages also in connective tissue, because connective tissue, for example, is not too packed. Sometimes we have this irregular connective tissue that doesn't have a lot of matrix, if you will. I'm sorry, not matrix, but fibers, if you will. And because of that, it gives it a good kind of area where they can, you know, continue to move through connective tissue. But you find those in connective tissue, and you also find those in the lymphoid organs. And when they find, for example, a pathogen, they will take the antigen to the T cell. And the interesting thing with a macrophage is not only does it activate the T cell, but it also activates other macrophages, right? And what it does is actually activates all the other macrophages, and these become what we call activated macrophages. And they become highly phagocytic, right? And again, the key word here is recruiting, right? Why would you present it to the T cell? You're recruiting, again, more defense, and more defense. And that's what you want to do, is really mount a good response. Because again, it's an invasion, right? We got to put the threat down. So B lymphocytes do this as well. Again, this is antigen presenting cells, and the B lymphocytes do this, right? Okay, so they actually present the antigen to what we call a helper T cell. Okay, and what a helper T cell does is it actually presents this to T cells, right? Normal T cells, naive T cells, and actually regular T cells. So the helper helps, right? So it's a B cell presents the antigen to a helper T cell that presents those to other T cells, right? Right? So we see that there, right? Helper is the go-between, if you will. And that's how the B cells really work. Okay? So this table actually puts down B cells and T cells and, you know, the similarities and differences there. So let's take a look at B cells, right? When they actually target an antigen, right? When they actually interact with an antigen, what do they do? Because again, we know that they present them to helper T cells, but how do they participate in this response, right? Remember that they're the humoral response, right? And when you hear humoral response, B cells, you always think of what? Antibodies, right? Because that's actually their main job. Yes, they're going to present it to helper T cells. Yes, that is a job that they do. It's a really important job. But the other really, really important job is to actually make antibodies, right? Antibodies. So sometimes they do this through a couple different things, right? Okay. And we'll see how this works here in just a second. But when we see B cells get activated, that's when the antigen binds to the surface receptor. We call this cross-linking, but that's where the surface receptor binds to the antigen. And what it does is it endocytoses the actual receptor and the antigen that is binded to. And then it triggers an activation of the B cell. We call that clonal selection, just the same as the other, what we just talked about. And then those B cells will turn into what we call effector cells. Okay? And what kind of effector cells are B cells turn into? We call these type of effector cells for B cells plasma cells. Right? Plasma cells. So those plasma cells are the ones that actually synthesize, make, and then secrete antibodies. And that's what they do, right? Those antibodies and specific antibodies are at a rate of 2,000 molecules per second for 4 to 5 days, and then they die off, right? Those plasma cells then die, right? Where do these antibodies circulate? Again, blood, lymph. They bind to the free antigens. They temporarily inactivate them, and then also mark them for destruction by other immune system cells, right? Okay? That's what they do. And so you see this here. This is part of figure 2111, where you see an antigen binding to just one type of B lymphocyte, right? This is the one here. I'll draw a square over it, right? Notice that those antigens bind to that one there. I want you to notice something different, right? Something very interesting here. One of those B lymphocytes has green colored receptors. One of them has purple colored receptors. One of them has a blue colored receptor, right? Everybody see those three there? And notice that only the purple one is the one that is binding to the antigen, right? Why is that? Think to yourself, see if you can answer that. Why do you think that the purple one is the only one binding to the antigen? Well, it's all about the antigen's receptor, right? The antigenic, what do we call that? Determinant. Right? The antigenic determinant, or that part of that antigen that that particular surface receptor of the B cell binds to. Right? Notice that the blue and the green aren't binding to the antigen because that's not the same exact receptor that they bind to. Remember, that the adaptive immune system is about being specific, right? So we know that that particular B cell is specific to those particular antigens. Okay? And what it does is it actually undergoes clonal selection. And again, I just mentioned that clonal selection is about that B cell between those two steps endocytosing, taking the surface receptor and the antigen in, causing an activation of that B cell, which we call clonal selection, and then those cells then produce then the effector cell, and we know that effector cell, those effector cells are called plasma cells. Okay? I want you to notice something here, that we have one plasma cell there, another plasma cell there, another plasma cell there, because they secrete antibodies. Okay? But one plasma cell doesn't look like the rest. What does that plasma cell do? It actually is a memory cell. Okay? It's primed, it's ready to go, and knows what happened and knows how to produce antibodies for that particular type of antigen, and it's kept behind. It doesn't participate in the attack, if you will, of this type of antigen. It stays behind. The rest of them do. The rest of them produce then, antibodies. Right? Which are here. Antibodies. But the B cell does not. The memory B cell does not. All right? So that's what we see there. Memory cells give us immunological memory. Memorizes the immune response to that particular antigen, and then that helps us for future exposures to that antigen. Now, what's going to happen if we get a second exposure, right? Well, look at the second exposure, right? Sometimes can be years later, weeks later, months later, any time after that, right? Well, let's say it's 10 years later or 30 years later. When we interact with that same type of antigen, the same exact one as we just saw, there's a big difference, right? Yes, it gets clonal selection. Yes, it turns into plasma cells. But look how many plasma cells get produced versus our last example, right? Many plasma cells get produced. And also we see memory cells get produced in greater numbers. But it also means that we produce a lot of antibodies. A lot of antibodies get produced. And those antibodies produce in great amounts. So taking a look at that, right? Taking a look at that comparison between primary exposure and secondary and so on, subsequent exposure, right? So when we see the first time primary exposure, it takes about 3 to 6 days to mount the antibodies, or the antibodies to actually get created get made and get secreted, right? And then antibody levels peak in about 10 days. Levels then die, they start to die. Infection is, maintained, it's eliminated, the threat is eliminated, and we move on from there. Let's see what happens in the secondary or any type of exposure past that. But let's say it's the second time around, right? Those memory cells stick around and they interact with the antigen, the same antigen again. But instead of days, that 3 to 6 days or whatever, we have the response within hours. 2 to 3 days is when we see the peak, much higher levels, and the antibodies can stay for weeks to months and not just days. So we have a greater, stronger immune response, which is a very interesting and very special type of immune response, and this is only found in the adaptive immune system. Okay, so this gives us a great example of that. Here's our first exposure here to the antigen. Here in a couple days we have our peak response. It's about day, again, day 10 here, right? We see our level there, our peak level, and then it starts to go down. But let's say we expose ourselves again, right, here to antigen A, right, our second exposure. What's going to happen here? Can everybody see the antigen level? I'm sorry, the antibody level there, production, how big of a difference it is between right here and right here, right? Huge, huge difference, right? Huge difference. Now, in the passive, right, passive portion of humoral immunity, this is actually where we actually can interject antibodies already made, right? So not the antigen where we get our body to then make the antibodies, and then those antibodies destroy the pathogen, or we take pieces of a dead bacteria and stick it in there, so then we have an immune response. No, what we mean here is that the passive humoral immunity means that we just stick in the antibodies already, right? So there's two major ways this can actually occur, right? Okay, we can do this naturally also and artificially. And how do we do this naturally? How does this work in the natural sense, the innate sense? Antibodies can be delivered in a special time in a special place, right? Right? If you're a fetus, you can get it through your placenta of your mom, right? So you get your antibodies from your mom directly, okay? Or if you're an infant, you get those through breast milk, right? Through breast milk. And in fact, that's why it's crucially important, right, for mothers to undergo breastfeeding, at least for the first couple days, that colostrum, because one, it provides all these wonderful nutrients, but number two, it provides these wonderful immune system cells, like antibodies, for example, which are secreted by, the immune system cells, but getting these into the body so that the body can actually use these for defense, right? Even though you don't breastfeed, that doesn't mean that you have a lower immune system. It does mean that you can get these from other places, but it does, as an extra additive, an extra enhancement to your immune system, right? These antibodies directly into either the fetus or the infant, right? So we can do this artificially too. We can actually interject, serum, right? Okay, into the bloodstream. And we can actually interject antibodies into the body, right? And this is, for example, if you have an infection, sometimes you can put antibodies directly in there if your immune system doesn't respond to a particular type of foreign pathogen. And some of these things, for example, like, infections like yellow fever or malaria or even Ebola, they're developing the antibodies or in synthesizing, making those antibodies in a lab, and then can interject those antibodies into you if you develop an infection from one of these pathogens, and that can actually help, your immune system out, right? So we can do this artificially. Okay, so this kind of summarizes, those two different types of humoral immunity, the active versus the passive, right? And how those become in the active if you get those by nature, right? Again, this is a normal childhood type of experience, right? The natural one. This is also childhood and adulthood, right? Vaccines, right? And then passive natural, right? Getting the antibodies from the placenta of the mother or from the breast milk. And then artificially, you can actually stick a needle into yourself, into the bloodstream, stick those antibodies in there. And again, they'll still help you fight the infection in question, right? All right, so let's talk about antibodies really quick. I'm going to kind of breeze through some of this. Some of this is not, I don't want you to go into a lot of detail with some of this stuff, so we're going to kind of go past this point, right? Antibodies are called immunoglobulins, by the way, and we see the initial IG, right? And so depending on, IG stands for immunoglobulin, right? And that really stands for antibodies. And so the letter represents the different types of antibodies that you have there, right? Immunoglobulins, right? And these are the ones secreted by those plasma cells, right? Those B cells, right? In our body, right? And there's actually 5 classes. You just need to know that there's 5 of those classes, but you don't really need to go into as far as those classes are concerned. I wanted to talk really quick about the structure. There's a little bit more to talk about this than, but I just want to kind of keep it really brief, right? Antibodies will usually have a shape kind of like this, right? We call this a T antibody, T-shaped antibody, or they have this kind of Y structure, if you will, and that's the Y-shaped antibody. They can come in 2 different forms, the T or Y, and this line represents all the amino acids bonded together, and they actually form this shape because they have these bonds that you find here and here, and we call these disulfide bridges, where there's a sulfate group in each of those amino acids causing this binding to occur and having this shape occurring, right? And again, there's a lot of extra information here that I don't want you just to look at, just to know that there's a Y-shaped antibody and a T-shaped antibody. The amino acids bind by disulfide bridges. That's all I want you to see. And I just want you to look at this figure. We're not going to study this figure. This figure you also see in the lab book, but you see that these yellow lines, yellow structures represent the disulfide bridges. And that really was what makes the protein its shape, right? We see the typical Y-shaped antibody there with the disulfide bridges making the Y shape. And if you notice, that there is a line here, right? That is your amino acid, right? Your amino acid forming this shape here. Amino acid forming that shape there. Amino acids forming that shape here, and then they all get binded together, right, by creating that Y shape. And lo and behold, that's a shape, right, that binds to then, right? Here's another structure just kind of showing you that each color represents a different amino acid chain, right, a different peptide. Call this a peptide. And so you got a blue one, you got a black one, a yellow one, and a red one in this computer image here, and this is your typical Y-shaped antibody. Okay, here are the 5 classes. Okay, all starts with IG, immunoglobulin A, M, D, G, A, right? And again, we talk about, if they're a monomer, or dimer, or pentamer, and you don't need to know only this, right? So we'll kind of, pass by that. And that's a table that you see the different types of shapes, right? There can be different types of shapes, and that's antibodies that you see there. And again, that's a little bit more detail I want to get into there as well, right? Okay. Now I want to talk about their function though, right? So I want to talk about what they do, right? Okay, and how they do it, basically. Antibodies can go through these processes, right? They can do these things, right? They can neutralize, they can agglutinate, we call this agglutination, fancy word to say. In some cells can cause this clotting to occur, right? Where antibodies bind to one cell or one antigen and bind to another antigen, and they create this kind of net, if you will. And sometimes if they're cells, then they become clots, right? What we call precipitation, falling out of the solution, and then complements, right? Complement. Okay. So I want to, here's neutralization here, and I'm not going to talk about this. I'm not going to discuss. I want to go this figure here because it actually shows all four of them, right? All four of them, and talk about the each one of these here, right? Here's neutralization, right? This is our first one here, neutralization. And neutralization means to what? Neutralize, right? Neutralize the threat. And what we see here in our example in the yellow is a virus, right? Here's our virus, right there in the yellow. And what we see in the blue, right, are your antibodies, right? Here's our antibodies. And you can see them binding to the surface of the virus. And what they're binding to is not the virus itself, but the surface receptors, which are considered antigens, right? Those are antigens. They bind to the antigen because they have the right shape just for that antigen, bind to them, and neutralize the threat. They prevent this cell, this virus, from entering a cell. So this would be our typical cell, and it prevents that from happening. So it neutralizes it, okay, temporarily holds it there, prevents it from binding to cells, and then also marks it for destruction, right? So this, antibodies surrounding this virus tells the other immune system cells, hey, we need to destroy this. This is not part of our body. Agglutination, you can see this here occurring. So this is one antibody there, right? One antibody can bind to the antigen on this red blood cell, and that 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 red blood cell, and form this netting, if you will, right? Where cells get binded together, and we call that agglutination, right? Agglutination, like clotting, if you will. And that's what you see, a clot, in fact, there, right? A clot. And precipitation basically means that these antigens that are found that are soluble can actually enter into cells, if you will, all right, enter into fluid, get trapped or binded to antibodies, creating this kind of pattern here, if you will, and that pattern can actually cause precipitation or the settling out, right? The settling out of those antigens, right? So they bind them together much like neutralization, and then they settle out of the solution. So they run and they clump together basically as well. And this actually is very interesting, the complement activation, because complement activation, when the antibody bonds to the antigen on a surface of a cell, right? So we see this surface of a cell here, right? This is our cell surface right here, okay? And we see the antigen there in the orange, and we see the antibody binding to the cell surface, right? What can happen is that the antigen will tell the complement proteins, right, which are part of the second line of defense, complement proteins to come and then start building a pore, right? Start building an integral protein, and what this does is it creates a pore and allows a hole to be made to cause then fluid to rush out of a cell, right? Rush out of a cell. Okay, and what's the effect of that? If this was a cell surface of a bacteria, it causes that bacteria to what? Explode, right? Lyse out, break open. Okay, which is very interesting that it does that, right? Okay, I think we're done here.